Okay, so today I'm going to talk about glycolysis. Just wanted to say that I'm not a professor, so if I say anything wrong, just correct me and I'll write the correct answer in the description box or whatever I said wrong. Yeah. Okay, so let's start with some basics. Glycolysis is the breakdown of sugar. So, sugar, which is glucose. So, glucose will diffuse from, so glucose will be available in blood, or it will be there, and it will diffuse from the blood vessel into the cytosol of a cell, and this process takes place in the cytosol in all cells, especially erythrocytes, muscle cells, liver cells. Yeah. And it's also the breakdown of sugar because we want energy. So the overall equation also is glucose plus, but you can just take a look at it, but you'll understand it more after this video, hopefully. Okay, so intermediates / metabolites and enzymes of glycolysis. So an intermediate is really any molecule that will take place in this breakdown, in this pathway, and it's these 11 metabolites. And glycolysis just describes how glucose will go from glucose to this, and then to this, and then to this, and then to this, etc., until you get to pyruvate, which is the end goal. Just wanted to say that there are also enzymes that will help in converting these intermediates. But a kinase, and I think this will come up later too, a kinase is an enzyme that transfers phosphate between ATP and a substrate. So please remember that. And glycolysis is the process from glucose to pyruvate. Yes, this one is also really important. Glycolysis gives energy by substrate level phosphorylation. So energy can be produced by two ways. It's substrate level phosphorylation and also oxidative phosphorylation. But glycolysis is substrate level phosphorylation, and I will tell you what that means in the next slide. But it doesn't generate a lot of ATP. Okay. So, glycolysis. Let's start with the first thing. Here we have a glucose molecule. We will start with a glucose molecule that looks like this. And the first step would be that glucose will go from the blood vessel and it will go inside to the cytosol. And when that happens, there will be an enzyme called hexokinase that will convert or phosphorylate glucose to glucose 6-phosphate. And yes. Okay, I just wanted to say that hexokinase is the one that is most common in the muscle cells, and glucokinase is the one that is most common in the liver. Yeah. Okay. And in this reaction between glucose and glucose 6-phosphate, we will use magnesium. And another important thing is we will use ATP. So we will actually we will use ATP in order to get ATP from glycolysis. And in step number 1, that will happen. And this also makes the first step of glycolysis an irreversible step, and it's a rate-limiting step. So this step will determine how fast the reaction will go, or how fast glycolysis will go. Yeah, and it's the first priming reaction, which means that it will invest ATP and phosphorylate glucose. And we phosphorylate glucose because we don't want glucose to flow out of the cell. So when it becomes a glucose 6-phosphate, it will not flow out, and also this will decrease the concentration of glucose in the cytosol, which will lead to a concentration gradient, and more glucose can enter the cell from the blood vessels. So that's step number 1. Step number 2 is just converting glucose 6-phosphate to fructose 6-phosphate with the help of phosphohexose isomerase. So it's just a conversion because it's either easier to then phosphorylate the molecule. Then we come to step number 3, and again, this is a really important step. This is a irreversible committed step. So when we come to step number 3 and it happens, this is how we determine that glycolysis will happen and not any other reaction that has the same intermediates. So what happens here is that we have the fructose 6-phosphate. So on the 6 carbon we have a phosphate right here, and what will happen is that we will use another ATP here. So we will invest another one, and also have a magnesium, and we will produce fructose 1,6-bisphosphate. And this is really important because now when we go from fructose 6-phosphate and we go to fructose 1,6-bisphosphate, so one here and one here, this will make the molecule really symmetrical and we can cleave it, which will aid in step number 4, which is the cleavage of this molecule by the help of aldolase. And then we will produce glycerolaldehyde 3-phosphate and dihydroxyacetone phosphate. Glycerolaldehyde 3-phosphate, you can see that it has an aldehyde, and 1, 2, 3 on the 3rd carbon we will have a phosphate. And this is how we get the G3P molecule, which is this one. And then we will also get a dihydroxyacetone phosphate, and when we get a dihydroxyacetone phosphate we will have a ketone group. And what we want is actually so this one, the G3P, will be in lower concentration because this one is because of the aldehyde group. We want to produce more of it because that's the one that's easier to oxidize. Okay. Now, what I've described is the preparatory phase, and we have right now minus 2 ATP. We have invested 1 ATP here and 1 ATP here. And the two most important steps here is step number 1 and step number 3. Just remember that. They are the two irreversible steps of the preparatory phase. Then we will go to the payoff phase. And here we will go from the glyceraldehyde 3-phosphate. Now just remember that we have the two molecules now. We have this one and this one. So 2 molecules that will go through the payoff phase. So everything will be doubled. Okay. So the G and we will use G3P for the molecule. So glyceraldehyde 3-phosphate will become 1,3-bisphosphoglycerate by the help of this enzyme. And this is really important. I want you to remember that in step number 6, we in glycolysis we have to have NAD+. We have to have it because NAD+ is the molecule that will accept 2 hydrogens from glyceraldehyde 3-phosphate. So it will take a hydrogen and become NADH. And this is really important because when that happens and we get a 1,3-bisphosphoglycerate, really what 1,3-bisphosphoglycerate means that on the 1st carbon and on the 3rd carbon we will have a phosphate group here and here. So this is so remember this because I will talk about lactate and why lactate is produced. So remember this. Okay. Then we will go from 1,3-bisphosphoglycerate to 3-phosphoglycerate, and we will do that by phosphoglycerate kinase. Again, a kinase means the transfer of a phosphor between ATP and substrate. So here will be our first ATP forming reaction. And this is what substrate level phosphorylation is. It's basically you have a molecule, then you add a phosphate to it and you get ATP. That's substrate level. And yeah, so that's here you will get your first ATP. Then we will go from 3-phosphoglycerate to 2-phosphoglycerate. So basically the phosphor group will go from the third carbon to the second carbon. And you can see that here. So from the third carbon to the second carbon. And it will use this enzyme. And then we will go from 2-phosphoglycerate to phosphophenyl pyruvate, or PEP. And we will do that by the help of enolase, which is a lyase. And this enolase will remove a water from the 2-phosphoglycerate, and it will produce phosphophenyl pyruvate. And you can remember this, so we have to, in the colloq, we will have to differentiate the molecules. Like they could give you this molecule and then you have to name it. So how you can differentiate between this one and this one? Well, PEP will have a double bond here, and 2-phosphoglycerate will not. And that's really logical because in enolase, which is a lyase, when you remove water, it will form double bonds. So that's from previous lectures. And then we will go from PEP to pyruvate, and we will use pyruvate kinase. Once again, a kinase, transfer of phosphor, so, or phosphate group or whatever. And we will form ATP once again by substrate level phosphorylation. And we will use a magnesium. Really, what's important here is, and I will talk about this really soon, step number 1, 3, and 10 are irreversible. That's one thing. Secondly, okay, and about the ATP, we'll we'll get to it. Anyway, what reactions are irreversible and what's their role? So as I said, irreversible reactions in glycolysis are 3, and it is reaction number 1, 3, and 10. So in reaction number 1, we phosphorylate glucose, so glucose can diffuse back into the blood vessel. Irreversible step and rate determining. Step number 3, irreversible committed step, gets phosphate on both sides of the molecule. It's good for step number 4 because we can cleave the molecule and we can get similar molecules. And step number 10, which is the second production of ATP, is also irreversible. Oh yeah, I forgot to mention that enolase can be inhibited by fluorine, so a heavy metal. Okay, so the enzymes for reaction number 1, 3, and 10, they are allosterically regulated, so they can have activators or inhibitors. And a good thing to remember is that activators, okay, so we have to remember that glycolysis, we want ATP, right? But if we already have ATP, then we don't want glycolysis. So a high concentration of ATP will be an inhibitor. So so we have hexokinase. So if we have ATP, that will be an inhibitor. But if we have AMP or ADP, that will activate glycolysis, because if we have AMP and ADP, then yeah. And for phosphofructokinase 1, which is the enzyme for step number 3, if there is AMP, ADP, and ribulose 5-phosphate, that will be activating the molecule. But if we have ATP, citrate, or PEP, then that will inhibit the pathway. And then we have pyruvate kinase. And pyruvate kinase will be inhibited or activated by PEP, fructose 6-phosphate, and inhibited by ATP, acetyl coenzyme A, and alanine. Okay, let's do some ATP calculations. So, when we do calculations, I like to look at this one and this one. So we look at the ATP. Can we find ATP somewhere else? Yes, we can find ATP here. We can find ATP here. And then also we have this guy right here. Okay, so let me just, and this we will talk about in electron transport chain, but basically 1 NADH equals approximately 2.5 ATPs. Yeah. So here in the preparatory phase, we have actually used ATP because ATP has been put in, and then instead we've gotten an ADP. So here we have lost 1, and here, so here -1, -1. So far -2 ATP. And here we will have 2 ATPs generated. Okay, we'll have 2 ATPs generated because this reaction happens 2 times because we have 2 molecules here. So therefore we will have 2 ATPs here and 2 ATPs here. So that's 4 ATP. So if we write everything so far, -2 ATP or -2 plus 4 ATP, or let's just say +2 because +2 here, +2. So that would equal 2 ATPs. And then we cannot forget about the NADHs. So we will get 2 NADH, which will equal 5 ATP. Okay, so this is important. If glycolysis is anaerobic, that means that we will only get 2 ATPs. But if glycolysis is aerobic, meaning it will go through the citric acid cycle and through the electron transport chain, that means that we will get an additional 5 ATP through the NADHs, because the NADHs will go through those pathways and they will generate ATP. So, and this is what I've summarized here. So just remember that. 2 ATP invested in preparatory phase, 2 ATP produced plus 2 NADH. This is the net ATP plus 2 NADH produced, 7 ATP if it's aerobic glycolysis, but if it's anaerobic glycolysis, then it will be 2 ATP produced because the NADHs will not be used. So 7 ATP if aerobic. If it's anaerobic, then only 2 ATPs. These will not be helpful. Okay. Next slide. So, when we have produced pyruvate, what will happen to it? Okay, there's a couple of things. If there is oxygen, i.e. it's aerobic glycolysis, meaning it will go into the electron transport chain, then pyruvate will turn into acetyl coenzyme A and then go through the citric acid cycle and then electron transport chain. But if this happens anaerobically, meaning there is no oxygen, then pyruvate will turn into lactate. Why would it do that? Okay, so here we have pyruvate. We will have lactate dehydrogenase, which is the enzyme that will transform pyruvate to lactate. This reaction is really important because it will generate NAD+. And remember when I told you that NAD+ is really important? See, it's really important in step number 6 because this is the molecule that will take up hydrogen. And And if it does that, then glycolysis can happen again and again and again, right? So we need NAD+. So when we form lactate, we will form NAD+, and then NAD+ can go into glycolysis, and glycolysis can give us ATP, and we have now energy. And this lactate thing will happen during fast exercise, for example. If you want energy really fast, then that's one. Okay. Anaerobic glycolysis. So this is the overall equation. Why does it happen? Well, the cell produces ATP at a higher rate than oxygen can be supplied to cells. So this is what I just talked about. Like intense exercise, the cell needs a lot more ATP, and oxygen cannot be supplied as fast as ATP is used, and so we go back into glycolysis. Okay, so negative features of glycolysis, it is basically this. Slow production of ATP, accumulation of lactate leads to fatigue and soreness of the muscle. Lactic acidosis, it can also lead to lactic acidosis, so a high amount of hydrogen, which decreases the pH, and this leads to enzymes that produce ATP get inhibited function or worse function, or I don't know really what to say about something like that. But we have a bicarbonate buffer system, which will try to maintain pH. So hydrogens can increase, but the buffer system will try to get the blood pH to 7.35, 7.45. If lactate is produced, then pH will be lower. Cancer increases lactic acid production, and lactate can lead to immune suppression and also lactate-induced secretion of VEGF. VEGF is a protein that results in the formation of new blood vessels. And so basically, if you have cancer or cancerous cells, and they also have to supply themselves with blood and nutrients. So what they do is that they need new blood vessels. And lactate will induce production of this or secretion of this protein or hormone, I don't know what it is, but it will secrete this VEGF and angiogenesis, I think it's called, like blood vessels will grow, and the cancerous cells can supply themselves. Okay, that was it for glycolysis. If you have any questions, just email me. Okay, have a good day.